Hello, welcome back to the development part one videos. We just got talking about how genes can be make specialized cells, and this slide just talks about how it would be not very good for a cell in the liver or in the lens to produce albumin, which is a liver enzyme, but the eyeball doesn't need that. So that's all that slide's talking about. This slide just talks about a few vocab words. We talked about totipotent. Those are cells that have all the types of genes and can become any other type of cell. But we have a few other words, pluripotent, which cells that can give rise to make up any other type of body cell, but no embryonic stem cells. Embryonic stem cells are considered pluripotent, but it can't produce any of the extra embryonic stuff like placental cells or anything like that. And then multipotent, these are cells that can turn into any other type of cell, but are more limited than the pluripotent. Adult stem cells, ones that you find in your spinal cord, can be considered multipotent. This slide just kind of shows a little bit more about the development where you got pluripotent cells here, and you can see how a frog embryo is not fully committed yet, and then eventually those terminations happen. This graph just shows if you're going to do some sort of nuclear transplants like they did with the sheep dolly to make a clone essentially, but you don't have to be a clone. It can just be implanting the nucleus into an embryo, any embryo. And if you do it early enough, you get a pretty high success rate of full healthy development. That's because the earlier you do it, the more totipotent cells are, or pluripotent they are, which is better. Determination is just talking about when cells are determined to be placed in a certain part of the organism. If this gets messed up, you can get some weird stuff where this is the normal thing here. You get, tail or the head parts, and then you might get some parts where the head parts are imposed onto tails and things like that, or the two tails and stuff like that. This is a slide just showing about how you can get certain cell layers in the blastocyst, which happens after gastrulation to make new stem cells, which can turn into all kinds of cells. This talks about how determination occurs. How do we determine what cells are going to make what parts? Two things. You got cytoplasmic determinants. These are things actually in the cytoplasm. And then you have induction via signals of neighboring cells. So a different cell next to it might induce a certain cell to become something else. The little triangles and little circles here show cytoplasmic determinants. In an unfertilized egg, you can see how that's already being kind of shifted. You got certain determinants here, certain determinants here. So after fertilization and you get two different cells, you can see the cells are not identical. These cells are going to go off to be something. These cells are going to go off to be something different. That'd be like cytoplasm determinants helping. Essentially, what's in the cytoplasm can actually help determine what the cell becomes. This slide kind of shows a little bit about the inductive signals. This cell is putting out some sort of signal molecule that can be picked up by the signal receptor, making this cell become specialized in some way. Initial developments are usually governed by cytoplasmic determinants. That's all this is saying here. Drosophila, which is a type of little fruit fly, has development a lot like ours. And it's actually easy to subject to genetic manipulation because they have, relatively more simple cells. And this is the life cycle of one of those little flies. And this is talking about pattern formation. Some cells are going to be the head, the thorax, the abdomen. And it's talking about how certain, cytoplasmic determinants or neighboring cell induction signals can be used to help determine that. Maternal effect genes. These are genes called either bicoid or nanos genes, and they're found in the mothers, and you can see how they work here. Bicoid genes are going to be carried to the anterior, nanos are going to be carried to the back. Then you're going to get some other secondary genes called hunchback genes or caudal genes that are going to be activated by the bicoid or nanos. Caudal or tail, hunchback or front. If these get messed up, you can get tails in the wrong spots or heads in the wrong spots. And that pretty much, in these slides, I'll go through that. watch